I'm Dr. Paul Harch. I'm a clinical associate professor of medicine at LSU School of Medicine here in New Orleans. I'm also the director of the Wound Care and Hyperbaric Medicine Department. And my primary specialty is emergency medicine with a subspecialty in undersea and hyperbaric medicine. It is the undersea and hyperbaric medicine that has yielded the most productivity in research in my career. And my interests are centered around hyperbaric oxygen therapy applications to acute and chronic brain injury of all different types. Over the past 20 years or so, based on some seminal findings we made in treatment of our divers here in New Orleans, both commercial and sports scuba, we found that we could apply a lower dose of hyperbaric oxygen to residual brain injury in these divers. And that application was able to be generalized to multiple other forms of brain injury or causes of brain injury. And amongst the most responsive of all of the diagnoses, traumatic brain injury has stood out. In chronic traumatic brain injury now, we've been able to show in both humans and animals that even years after the traumatic event, you can reclaim functional outcome.
So I'm Dr. Nicolas Brezan, a faculty at the School of Medicine. I'm in the Department of Neurology, and I'm also the head of the LSU Neuroscience Center. My interests are trying to unravel the intrinsic mechanisms of the brain when confronted to injury. So during the years, we have investigated what happens, for example, in experimental epilepsy, what happens in stroke, what happens in PTSD-like conditions, using the brain circuitry as a model to try to figure out the foundations of the behavioral dysfunctions. In doing this, what we were able to identify is a novel family of molecules that the brain produces. The main one, we coined the term neuroprotectin D1 because it's a major regulator of the initiation of injury at the level of inflammation and a protector in the long term of the consequences that happens in brain circuitry. We are over 100 neuroscientists at the LSU Neuroscience Center. At present time, we are 15 different teams, including my own, and what we have built is a web of interdisciplinary interactions, because this is the only way that we are going to conquer brain injury, brain diseases, and consequences of brain diseases. We began a very productive and enjoyable collaboration with Dr. Paul Harch several years ago, first thinking about stroke and the penumbra, and a few years ago thinking about recapitulating in models what happens when the heart stops pumping blood to the brain. And we were able to identify the intimacy of key mechanisms in this dysfunction of the brain. We both believe that there is a tremendous potential synergy between what Dr. Harch is doing and what we are doing on the basic front, because there is a variability in how each of us respond to injury. How our brain responds to injury today, we know that in part is due, for example, to genes, and there are gene susceptibility brains, and it's a whole new world of the neurosciences, of the clinical neurosciences. We only know about 2% of the brain genes. All the others have not been identified. 2%, I should say, of the DNA of the brain. Thinking in this trend of thoughts, we collaborated, and I wanted to mention as an example a paper that was published in 2008 together with Dr. Harch and our team, where we identify DNA-related cell damage to the brain under these conditions. And this is going to be the way to really move this field forward, because a key word that we bring to the table is neuroprotection. And we have neuroprotective molecules, neuroprotective substances that could actually make the changes, the therapeutic changes that Dr. Harch is achieving of a more sustained manner.
We have difficulty containing our excitement over the development of these parallel but independent pathways in Dr. Bazan's lab and with our group headed by Dr. Keith Van Meter. Essentially what we are finding is that from the human side, which is rooted in the animal experience, hyperbaric oxygen has extremely powerful anti-inflammatory effects and effects on the white blood cell component of inflammation after acute injury. What Dr. Bazan's lab has found is that their neuroprotective agents also are potent anti-inflammatory medications or drugs, agents that are acting on inflammation in a more generalized way, as well as on white blood cell mediated inflammation. Considering that there are probably 1 million genes in the human genome, each of them coding for a different protein or enzyme, there are multiple agents and proteins and enzymes that are involved. The fact that their discoveries and ours are involving similar types of end effects, we think that the possibility for synergy here is inestimable, and we are extremely excited about that.
One very interesting issue that we have talked very often is the issue of anti-inflammatory. In medicine, we have a very large number of anti-inflammatory medications. Many of these medications, what they will do is delay what we can call resolution of inflammation. Because when there is an injury in the brain, there is all of this surge in white cells into the brain and damaging chemicals happening. Most current anti-inflammatories will delay for this to be resolved. And in severe traumatic head injury, what happens is that as a consequence of this, there is a irreversible, long-lasting injury. Our discovery of neuroprotectin D1 have demonstrated that what we are dealing is not only with the inflammation itself, but what we are doing is enhancing the resolution of neuroinflammation. And by enhancing the resolution of the neuroinflammatory process, we are actually achieving protection of the function and integrity of the brain cells. And this is very exciting because it opens a tremendous potential for many diseases. We tested, as you know, because we wanted to know if this would happen in the human brain. And we tested the hippocampus of patients that are in early stages of Alzheimer's disease, because we said, well, if this system is meaningful, number one, it has to be present in the human brain. Number two, it has to be somehow failing in conditions where we have clear pathology. And we were fortunate to be able to benefit ourselves from donors that provided their brains for research, both without brain pathology and a group of patients with early stages of Alzheimer's disease. And what we found is, as you know, Paul, is a 25-fold decrease in the signaling and in the molecule neuroprotectin D1. This is a paper that was published the year of Katrina in 2005 in the Journal of Clinical Investigation, and has given us the foundation to the thinking that this resolution of inflammation mechanism in the brain may be very important, not only to sustain the structure, but perhaps to inhibit the apoptotic cell death that inevitably happen after these type of processes.
You are so right on with your analysis and explanation of this. And the beauty of it is that we are talking about a generic process. Inflammation, as Dr. Bazan is illustrating, and their findings are present in Alzheimer's, in traumatic brain injury, in focal ischemia such as stroke, and in many other processes. And that is identical to what we are finding. What people have not realized is that regardless of the type of insult to the body, whether it's trauma, withdrawal of blood supply, withdrawal of oxygen, a toxin, an electrical shock, any type of insult, there may be idiosyncratic differences in the acute insult, but the second that the insult is delivered, the inflammatory reaction plays. And it's like plugging in a DVD. You get the same stereotypic reaction for every insult. So when you intervene downstream, which is what Dr. Bazan is doing and what we are doing, the effects on inhibiting inflammation are a generic type of approach. And that's the beauty of it, because the inflammatory process is in most of our acute and chronic diseases. What's also very important is what they are targeting are genomic types of effects and intermediaries that are acting at the DNA level. What has now been shown with hyperbaric oxygen therapy in the last decade is that the primary target of hyperbaric oxygen are likely the promoter segments of genes. In a very key experiment that was done 2 years ago on human microvascular endothelial cells, a single hyperbaric treatment was shown to activate over 8,100 genes. The genes that were upregulated were those that code for growth and repair hormones and the anti-inflammatory genes. The ones that were downregulated were the pro-inflammatory genes and the genes that code for cell death. And one of the things that hyperbaric oxygen has been so clearly shown to do in acute injury models is to inhibit cell death, much like the neuroprotective agents that Dr. Bazan's lab has developed.
Yeah, one of the exciting parallelisms between what Paul have found with the hyperbaric treatment and what we found in the human brain of patients with Alzheimer's disease is that in the hippocampus, what we did is we took then cells from the human brain, as you know, put them in a petri dish. These were brain cells in culture in a petri dish, and then we injured them. Not with hyperbaric injury like or treatment as you did. We gave them an injury, and then we found exactly what you found, but the beneficial effector here was neuroprotectin D1. The beneficial treatment in your petri dish experiment was hyperbaric treatment. Right. So that parallelism at the mechanistic point of view is very revealing. Yes. Because they may be converging on these fundamental mechanisms. Often in brain research and in brain damage, we talk about discovering the highway, the main road pathways for neuronal survival. And often neuroscientists said, let's make sure that we are not taking the service road to get there. I believe what Dr. Harch is doing, he is truly in the main highway, because this is a major effector system. And we believe, and there are many things that we need to do together, that the cellular and molecular parallelism might demonstrate enhancement of the action of both systems when we put them together in animals and in humans.
Yes. And that is the huge potential here, is adding 1 and 1 and getting 3, 4, 5, or 10. And along those lines, some of the very important work done in this was done by my senior partner, Dr. Keith Van Meter. And all of this, again, started with our discoveries
Absolutely.
in diving medicine, which is somewhat unique to this New Orleans area. We had a very unusual combination of our exposure to the commercial diving industry, the local geography, and our treatment of divers. And in 1984, he was able to resuscitate a diver who had extreme decompression sickness, drowned, and had a cardiac arrest here in the Mississippi River, right behind our local zoo. And after 25 minutes of cardiac arrest, the patient was put in a chamber, and in an error of the diving company, in their attempt to resuscitate them, accidentally this patient was given pure oxygen at 6 atmospheres. Usually the limit any human is given is 3 atmospheres. To everyone's surprise, after 25 minutes of cardiac arrest, this diver was completely resuscitated. And in fact, with the high levels of oxygen in the chamber, which were therapeutic to him, they were toxic to the inside tender who was tending him, who then had an oxygen toxicity seizure. The resuscitated diver then tended to the oxygen-stricken inside tender. The net result was a complete salvage. And 27 years later now, 28 years later, this man is a custom furniture fabricator, is married, has children, and came back to New Orleans this year. We imaged his brain. We cognitively test him. He has an IQ near 120. He's totally functional and happy. That spawned the resuscitation experiments of Dr. Van Meter, where now after 25 minutes of cardiac arrest in a swine model, he is able to resuscitate pigs from cardiac arrest. And in conjunction with Dr. Bazan's lab, they did the clinical work, or the basic science work, showing that it arrested the inflammatory process and cell death process in the brain. So again, the potential synergy of what we are doing and the neuroprotective agents have really huge potential.
You see, a few years ago, let's say a decade ago, well, let's put it in the early 80s, when Keith Van Meter made that observation, the neurosciences were a promise for medicine. Now, in 2011, I believe there are specific channels, concepts, technologies, knowledge that can actually be applied. It's no longer a promise. And this is the excitement of the times that we are living, because we can merge all of this wonderful work of Dr. Harch with Dr. Van Meter and the basic science. And this conjunction of approaches, I think, can only be of benefit to our soldiers and to mankind.
Oh, yes. And considering that inflammation is a ubiquitous process present in almost every disease, a therapeutic application or combination of therapies that target inflammation has the potential to treat a wide spectrum of diseases, not just the ones that we have mentioned here.
Absolutely.
So,
well, if you want to add something to the inflammation, because the leukocytes, when they get into the brain after stroke, they're beneficial.
mm-hmm.
They are there for an absolutely beneficial reason. They are part of the immune innate system, and they are part of a physiological response to cope with the damage.
Right.
But if the breakage of the blood-brain barrier is large, and you have a lot of them, and that response is exaggerated, is when neuroinflammation is triggered, and when we need to regulate the resolution of that inflammation.
Right.
And so this is exciting. We couldn't talk about this cellular and molecular mechanism the way that we are doing it now 20 years ago.
No.
And this is the time to do it now.
It is. It is. The time has come. The basic science has now caught up to the clinical experience, which has been slowly evolving over the years. And there's great potential.
And we are all here in New Orleans, actually, and we have a very good facility at the LSU Neuroscience Center, where the basic research is being conducted, and Dr. Harch and Dr. Van Meter have a clinical facility here. So I believe the region is poised to make a major contribution to something that had no alternative therapeutic solution up to now.
I agree.
Delighted, Paul.
I agree.
Delighted.
This is wonderful. Wonderful.
Absolutely.
So we have high hopes and expectations.